Chapter 24, the Immune System. In Chapter 24, we're going to look at the components of the immune system in vertebrates. The innate immunity is an immune system we're all born with. It is the first line of defense against pathogens or disease-causing organisms. Adaptive immunity, that's acquired over time. It involves certain components of our immune system, B cells and T cells, and antibodies. We're going to talk about that. And last but not least, we're going to talk about when our immune system attacks our own body, leading to several disorders such as rheumatoid arthritis, multiple sclerosis, type 1 diabetes, among others. Innate immunity. Alright, in the world we live in, there's a lot of little microorganisms that cause disease. We call them pathogens. The immune system is the body system responsible for protecting us against pathogens. There are two sets of defenses. Innate immunity is the first line of defense. It kicks in immediately after we've been infected. Innate immunity does not change. It's the same. It acts the same whether or not the pathogen's been encountered before or the first time. Invertebrates, all they have to protect them is innate immunity. And in these organisms, their innate immunity is based upon their exoskeleton and other barriers, physical barriers to penetration by pathogens. Inside the body, low acidic pHs contain an enzyme called lysozyme, which digests the cell walls of bacteria. And like us, they have immune cells, which are capable of cellular eating phagocytosis, which eat foreign materials. Vertebrates have innate immunity, but also adaptive immunity. So, in invertebrates, they have external barriers like the exoskeleton, an acidic environment. Secretions produce secretions, which contain compounds against micropathogens. We have mucous membranes, hair, cilia that line our trachea, which help filter germs and other things out of our lungs. If these external barriers are gone through or breached, we have innate internal defenses, phagocytic cells, white blood cells, natural killer cells, cells that attack pathogens directly. Defensive proteins like antibodies, interferon, and the inflammatory response, which speeds up healing. Vertebrates, all vertebrates animals have innate immunity. In vertebrates, innate immunity includes the skin, mucous membranes, which act as filters to keep and prevent penetration by germs. Interferons, these are proteins produced by viral infected cells, which protect other cells against viral invasion. Neutrophils are the most abundant white blood cell. They protect us by phagocytosis. They eat pathogens. Macrophages are another type of white blood cell. These are large phagocytic cells that are not restricted to the blood. They actually can leave the circulatory system and wander through the tissues, through the interstitial fluid. A special category of white blood cells are called natural killer cells, which specialize in attacking cancer cells and viral infected cells. And our blood contains proteins. It's a series of proteins which activated can attack and destroy pathogens. Alright, here's a virus. right here, and it infects a cell. Well, the actual infection of the cell by this virus causes genes to be turned on that produce interferon. Interferon is a protein which attacks viruses. When the cell is lysed or ruptured, the interferon may be picked up by other cells, which protects them. It triggers a response to protect other proteins to protect them against viral invasion. When we damage our tissues, whether it's a splinter or a scratch, it triggers a response called the inflammatory response. The purpose of the inflammatory response is to disinfect and clean infected tissues and limit the spread of infection. There's a series of events. When the tissue is injured, mast cells release signaling molecules. Macrophages also will release molecules that cause the blood vessels, the capillaries, to become leaky. And when that happens, the molecules that are released attract other white blood cells, such as neutrophils. Also, because the vessels are leaky, interstitial fluid rushes in. Now, the neutrophils digest the bacteria and all the garbage cell debris at the site, and that speeds up the healing response. Now, inflammation may be on a local or it may be a body-wide systemic. Sometimes the microorganisms that get into our blood release toxins which can be spread. Now, a bacterial infection that brings a systemic inflammatory response that can be very dangerous. It can cause a drop in blood pressure and can cause death. Adaptive immunity is called acquired immunity. It's a set of defenses only found with vertebrates that is activated only after exposure to specific pathogens. Now, adaptive immunity is specific for individual to individual. It's not like innate, and it depends upon what pathogens that individual has been exposed to. So, innate immunity is a very rapid response. It recognizes a broad range of pathogens but has no memory of infection. Adaptive immunity has a slower response but has a memory. It recognizes specific pathogens. Now, the thing that triggers adaptive immunity are antigens. Antigens simply any molecule that elicits an adaptive immune response. It's typically a piece of the pathogen. It's a molecule that is not belong to the individual. It's non-self. It can be anything that protrudes from the pathogen or any other particle that is not self but triggers a response. So when the immune system detects an antigen, there's several things that can happen. Number one, increase the number of white blood cells, either attack the invader directly or cells that produce antibodies, defense proteins. So antibodies are proteins found in your blood that will attach to only one specific type of antigen and neutralize it. So adaptive immunity has the ability to remember previous infections and be ready for it if that particular pathogen decides to strike again. So it can remember antigens it has encountered even years before. And the second response will be much greater than the first response. Now, the white blood cells responsible for adaptive immunity are lymphocytes. Lymphocytes are found throughout the body, in the blood and in the lymphatic system, and they provide specialized defenses Now, adaptive immunity can be obtained by natural exposure to antigens, but we can trick the immune system into an adaptive immune effect by vaccination or immunization. In a vaccine, your immune system is subjected to a harmless form of the antigen, or part of the microbe, which triggers an immune response, but you don't get sick from it. So antigens can enter the body like being infected with the flu virus, you get the flu, or getting a flu shot. The immunity is active immunity because the person's immune system will produce antibodies to that, whether it's a virus or a bacteria. And in cases of accidental exposure, you can be immunized with pre-made antibodies. This is called passive immunity. It's not active because your immune system doesn't respond to it, and immunity wears off as soon as those proteins break down and the body recycles them. The lymphatic system is the body system that is involved in immunity. It is a network of vessels, blood vessels, and special organs. Lymph nodes are part of the immune system. They're situated at specific points amongst the lymphatic vessels. The fluid that flows through the lymphatic system is lymph, which is similar to interstitial fluid. So what lymphatic vessels do, they collect excess fluid from body tissues and return it back to the circulatory system. So, lymphatic system does two things for us. Number one, it returns fluid to the circulatory system. Number two, it protects us against disease or infection. So, as this lymphatic fluid circulates through the lymphatic system, it collects toxins, microbes, parts of microbes, and transports it to the lymphatic organs. Macrophages are located in the lymphatic organs, which includes lymph nodes, and that's where these invaders can be engulfed and digested. And also, lymphocytes may mount an adaptive response from the lymphatic organs. Examples of lymphatic organs are tonsils and adenoids, thymus, spleen, and also MALT, mucosa-associated lymphatic tissue. That's found in the tonsils, in your digestive tract, and other parts of the body. Lymphocytes originate from stem cells in the bone marrow. There's two types: B lymphocytes, which once developed they mature in the bone marrow, and T lymphocytes, once they form, go to the thymus, complete their development there. So B and T cells are the main parts of adaptive immunity. So B cells and T cells develop from stem cells in the bone marrow. As immature lymphocytes, they go to one of two places to complete their development. Future B cells go to the bone marrow, future T cells go to the thymus, where they complete their development. From there, they'll move to other organs such as lymph nodes. B cells mount a humoral response. T cells mount a specific response. Okay. When a lymphocyte develops either in bone marrow or in the thymus, the cells will differentiate from other cells. They will synthesize a protein called antigen receptors, which is capable of binding one specific type of antigen. Once these proteins are synthesized, they're incorporated into their plasma membranes. Now, all the antigen receptors on the surface of a single lymphocyte are identical, and they recognize a specific antigen. And you have many different types of these cells in your blood and in your lymphatic system. So B cells and T cells provide a two-pronged attack. B cells participate in the humoral immune response, which protect you against bacteria and viruses found in body fluids, blood and lymphatic fluid or interstitial fluid. And they also secrete antibodies at the same time. T cells participate in cell-mediated immune response. They attack specific cells. The cell is infected with bacteria and viruses. They defend against infections. And they promote phagocytosis. They tell other white blood cells to eat the foreign cells. They also release chemicals that stimulate B cells to release antibodies. Alright, both responses are initiated when lymphocytes recognize antigens. B cells bind the antigens directly. T cells have to undergo an extra step to recognize antigens. Well, a couple of things to keep in mind. The antigens usually do not belong to the host animal. Mostly they're proteins or large polysaccharides that are attached to viruses or foreign cells. The protein coat of viruses, the parts of the capsules and cell walls of bacteria, and macromolecules on the surface cells of other cells, protozoans, parasitic worms. Those are all antigens. Alright, antigenic terminants are specific regions on an antigen where the antibodies bind. So we find antigen receptors on white blood cells, on B cells, as well as antibodies. And what they do is recognize specific antigens. Now, talking about antibodies, the specific region of the antigen receptor or antibody that recognizes an antigen is the binding site, and it's like a lock and key. An antigen will usually have more than one determinant. So you can have two different antibodies binding to the same antigen. Now, both B cells and T cells protect you by cloning more, making more of the same type of cells. We call this clonal selection. So when an antigen encounters B and T lymphocytes, you have all different types. Only one particular antigen will interact with only a fraction of those, not all of them. Now, once a lymphocyte is activated by the antigen and binds to it, they form a clone of thousands of cells that respond to that specific antigen. Now, some of those cells are effector cells. They jump to work immediately. The rest are memory cells. They lie in wait for future infection by the same antigen. Alright. So we have antigens bind to a B cell that has the correct receptors. Now, B cells with different antigen receptors do not respond. The selected B cell gives rise to a clone of B cells with the same receptors, the same antigen. Some B cells become effector cells. They jump to work immediately. Others are memory cells. They lie in wait for the next time. So memory cells last for decades. Effector cells only last a short period of time. Now, when we talk about the immune response, there's the primary response, which occurs upon first exposure to the antigen. and it's slower than a secondary response. And the secondary response is the second exposure, and it's faster and stronger than the primary response. The secondary response activates effectors in memory cells, but it happens faster than it does with the primary response. So this is a primary response to antigen X. First exposure takes about 7 days before there's enough antibodies to do anything. The secondary response, we have a heightened concentration and it takes shorter amount of time. But let's say it attacks the same, let's say pathogen but it comes up with a different antigen. Well, it still recognizes the antigen X, but it has a primary response just to antigen Y. Now, antibodies don't kill pathogens, they just sort of neutralize them. They help in getting rid of them, but they don't do it by themselves. An antibody is a protein molecule made up of 4 polypeptide chains. It looks like a Y, and the tip of each arm forms an antigen binding site that works on antibody recognition and antibody binding. There is a huge variety in 3-dimensional shapes depending on with different antibodies. That's why you have so many different lymphocytes and they react to so many different antigens. Looks something like that. Now, all antibodies involve a specific recognition. They recognize the antigen, they bind and attack. Once they attack it, they destroy it. In order for this destruction to take place, the active response has to work with the innate immunity. Alright, so the antigen-antibody complex, it works with phagocytic cells of the phagocytes of the innate immunity. Well, number one, the antigen-antibody complex neutralizes the pathogen. And then it causes a clumping response, where the antibodies clump with the foreign invader. And this causes it to form lumps, sort of like clots, which speeds up the phagocytic response.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least 2 binding sites, allowing them to form a clump of microbes by binding to 2 antigens on 2 different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell, The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.
Alright, so this illustrates the antibody response. Neutralization. By attaching to the foreign invaders, it neutralizes them, prevents them from infecting cells. Agglutination, the antibody can bind more than one and form a clump. This speeds up the process to get it ate, but it enhances phagocytosis by macrophages. Precipitation is where they bind to dissolved antigens and clumps them together. And by activating the complement system, the complement proteins can actually punch a hole into the foreign cell and cause its rupturing or death. So, in addition to phagocytosis, the antigen antibodies activates the complement system. And once activated, these complement proteins can attach to a foreign cell and bring about its destruction. Alright, antibodies are used in research, home pregnancy kits, and in treatment of cancer by designing specific antibodies that recognize cancer cells and carry toxins. They can bind to these cancer cells and poison them by using these toxins. And in pregnancy human kits, they use an antibody to detect a hormone which is excreted in urine called HCG. Adaptive immunity to a specific pathogen can be acquired by vaccination or natural infection. The HPV virus it's a common infection for adults. Usually there are no symptoms and your immune system gets rid of it. But in some individuals, the virus escapes the immune systems and also can, when it affects epithelial cells, it can cause cancer. Now, there are 2 vaccines out to promote immunity against HPV called Gardasil and Cervarix. Well, research has shown that the vaccines are effective, but little is known about how long vaccination lasts. Well, all you can do is measure the antibody level in people who've been vaccinated. Do a simple blood test to measure the level of antibodies, and you get an idea if a booster may be indicated. But according to this chart, it seems that the Cervarix lasts longer than the Gardasil. Alright, now the cell-mediated immune response. This is where the cells specifically battle the pathogens. This is done by cytotoxic T cells. Cytotoxic T cells are helped out by helper T cells. What they do is these helper cells can initiate antibody production and activate the cytotoxic T cells that will kill infected cells. Now, in order for a T cell to activate an immune response, there has to be an antigen present that will bind to the antigen receptor. And then the antigen must be displayed on the surface of an antigen presenting cell, such as a macrophage and B cell. Alright, let's look at an example here. A macrophage eats a bacterial cell and breaks it into fragments. Alright, now these antigens, there's other molecules of a special protein belonging to the macrophage called a self protein. which binds the foreign antigens and then displays them on the cell surface, creating a self-non-self complex. And helper T cells recognize and will bind to these complexes. So what happens, a macrophage eats the bacteria and breaks it down into smaller parts, the antigens. Self-proteins bind to these antigens and then are incorporated into the cell membrane of the macrophage. Now, the helper T cell will bind to this complex here, and what that does is the activated cell is now activated. It will release signaling molecules, and that will stimulate clonal selection of the helper T cell, producing memory cells and more helper T cells. And then at the same time, these stimulating proteins activate B cells, thus stimulating the humoral response and then stimulate the activity of cytotoxic T cells, which brings about the attack on the foreign invaders.
Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen. Correct. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. What kind of cell now responds to the antigen-presenting cell and activates other cells to attack the invaders? Click on your choice. Correct. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. What kind of cell now responds to the antigen-presenting cell and activates other cells to attack the invaders? Click on your choice.
So, the activated helper T cell binds to the foreign invader, the antigen-presenting cell, releases chemical signaling molecules that stimulate the B cells, and also signals the cytotoxic T cells. Cytotoxic T cells are the only T cells that kill infected cells. Once they're activated, They duplicate themselves, and they identify infected cells through a self-non-self complex. Well, an infected cell will have foreign antigens attached to its self proteins.
Now let's look in more detail at cell-mediated immunity. Cytotoxic T cells are the main warriors of cell-mediated immunity, which attacks body cells that have been altered by infection or cancer, or transplanted cells that are foreign to the body. An infected, cancerous, or transplanted cell displays foreign antigens on its surface. T cell receptors on specific cytotoxic T cells recognize and bind to specific antigens on cells. Cytotoxic T cells respond to this contact and to interleukin-2 sent out by helper T cells by producing the protein perforin. Perforin molecules are released from the cytotoxic T cell and attack the membrane of the infected cell, punching holes in it, which allows water and solutes to enter. The cell bursts and is killed. In this way, cytotoxic T cells deprive pathogens of their host cells so they cannot reproduce. Tumor cells are also removed from the body by cytotoxic T cells. In this way, cell-mediated immunity, carried out by cytotoxic T cells, destroys altered or infected body cells.
So any infected body cell that has a self-non-self complex signals the cytotoxic T cells if it has the correct receptors. So when it binds to the infected cell, it activates it. It synthesizes proteins such as perforin. What perforin does is like a bullet. It punches holes in the infected cell, and it promotes its death. So once the holes are punched in it, enzymes produced by the T cell will enter the infected cell and bring about its demise. And then that cytotoxic T cell will move on to destroy other cells infected with the same pathogen. So here we have a cytotoxic T cell binding to an infected cell. It binds to the self-non-self complex. It releases perforin to punch a hole, and then enzymes that promote cell death, apoptosis. Alright. Cytotoxic T cells play a part against protecting the body against cancer cells. About 20% of human cancers are caused by viruses. HPV causes cervical and anal cancers, hepatitis B, liver cancer, Epstein-Barr, which causes mononucleosis, can cause lymphomas, and one type of leukemia. AIDS, acquired immunodeficiency syndrome, results from infection by the HIV virus. It's killed a lot of people. And nearly as many people that's been killed by AIDS live with it today, or infected with HIV. Alright. Most infections and deaths from AIDS and HIV occur in other countries. Now, the virus that causes AIDS attacks helper T cells. And what happens, it impairs the cell-mediated immune response, it impairs the humoral response. What typically kills the person with AIDS is not the HIV, but opportunistic infections.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named, retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Alright, so AIDS is not curable. Although drugs can slow it down, there is no cure. The key is prevention. Another problem with HIV is the virus has a very high rate of mutation. And that makes it hard to treat AIDS because the virus is evolving resistance to the drugs currently used to treat AIDS. Alright, when the immune system turns against itself, that's when autoimmune diseases such as lupus, rheumatoid arthritis, type 1 diabetes, multiple sclerosis, and perhaps Crohn's disease. Immunodeficiency diseases are when the immune system underreacts, in which the immune system has been compromised. This can be due to cancer, exposure to radiation, or stress. Allergies are when your immune system overreacts to harmless antigens. Antigens that cause allergies are called allergens. Allergic reactions typically occur very rapidly in response to tiny amounts. Allergic reactions can occur anywhere. Nasal passages, your bronchi, your skin. Allergic symptoms result from a two-stage response: sensitization when a person is first exposed. Then the second stage begins when the person is exposed to the same allergen later. Allergen binds to mast cells. The mast cells release histamine, which causes the symptoms of the allergy. Antihistamines interfere with the action of histamine and temporarily relieve symptoms. Now, when an allergic response occurs over the whole body, that's called anaphylactic shock. That occurs when you've been in certain allergens with bee venom, penicillin, or allergies with peanuts or shellfish. It gets treated with epinephrine injections.